Hi friends, in this we are going to discuss about the metroglyph analysis. First we need to know what's the metroglyph analysis. It's a semi-graphical method for assessing the pattern of morphological variation in a large number of germplasm taken at a time. And this technique is developed by Anderson in 1957. And he has he done to investigate pattern of morphological variation in crop species. The main feature of metroglyph analysis are the analysis is based on first order statistics, first first order statistics. That means statistically more reliable. First order statistics. It's a more reliable and robust and very simple analysis. Analysis is possible from both replicated as well as and non-replicated. Data pattern of variability is depicted by the glyph on the graph. And to analyze this, there are some steps are involved, like selection of genotypes. First one, selection of genotype. Selection of genotype, and each material which is used for assessment of variability may include germplasm, lines, varieties, hybrids, and genotypes are selected based on their phenotype. And evaluation of materials, and the selected genotypes are tested in replicated field trials, observed and recorded for various quantitative traits, and mean values over the the replications for each traits are worked out. And final third step is second one evaluation, third one assessment of variability, assessment of variability. First one selection, second one evaluation, third one assessment of variability. And the semi-graphic method Anson used for the assessment of variability, which consists of following steps, like plotting of a glyph on the graph, that is small circle by which the position of genotype or line is represented on a graph is called a glyph. As we can see that here, a small circle which is pointed position of genotypes or lines represented on a graph. This is a small circle. This we can call it as glyphs. And these are based on x and y axis. X and y axis and x axis. And each line occupies a definite position on the graph called as glyph. And depiction of variation. Variation for remaining characters of the glyph is depicted by on the respective graph by rays. The character occupies definite position, ray position. Variation for each character is depicted by the length of ray. Thus the length of ray may particular character on which a glyph may be short, medium, and long on the index value of the genotype. Like I said that ray, if it's a glyph, then we can see that some of the rays, the variation is depicted based on the length of the rays also. And final construction of index scores. And the last one, analysis of variation. Let's come to study this graph briefly. Let's study this with an example. Like let us consider 35 genotypes of cotton. 35 genotypes of cotton and 5 characters. 5 characters. It's the genotypes and it's a 5 characters, like seed cotton, yield per plant, ball weight, number of balls per plant, ginning outrun, and the mean halo length. The yield and the balls per plants are highly variable characters which will be taken on x and y axis, and other traits are depicted on the graph like a yield and ball per plant. Here we can see yield and balls per plant. It's a highly variable, so we take this as a major one. So we depict it like this, and we can in this um, actually high, low, medium means high yield, medium yield, high yield, medium yield, and low yield, and less ball for low balls per plant, medium balls per plant, high balls per plant. By this um, Metroglyph analysis of 5 characters in 35 cotton types. In this we can say that the yields per plant and balls per plant, it's 2 characters, and this the glyph arrays shows that the different characters in three arrays are there. These show 3 different characters. If length of this array shows the remaining characters like ginning outrun, mean hollow length, and um, ball weight. So I suppose with this graph we can analyze that the sum of the coming to the here these 4 genotypes depicts that it has more number of balls per plant and also high yield. And coming to this one, it's a less yield and less balls per plant. Coming to here, it's a these are a medium yield and medium balls per plant. By this we can easily analyze the variation present in the genotypes. We can select we can select like required. If we require more, if we screen or select the best one, high yield per plant and more ball per plant. With this we can analyze we can study this thing metroglyphic chart, and its analysis is very important, useful in mainly 3 ways. Like it helps in studying pattern of morphological variation in large number of germplasm lines at a time. We can study these are all large number of sample can be studied in a large number of time. And the procedure is very simple technique and both unreplicated and replicated data, because analysis is based on mean values of a character, useful in classification of germplasm. And coming to demerits, main disadvantage is that in inclusion of large number of genotypes sometimes leads to overlapping of glyphs on the graph. If it becomes more number of genotypes, then it will becomes like a clumsy. We cannot overlapping, like if there is glyph, if there is glyph, if there is a glyph like this, then we cannot easily differentiate. Means we cannot sometimes there is overlapping, we cannot differentiate, we can study easily. And metroglyph, this is the main demerits of this. Metroglyph analysis has been used for assessment of variability in cotton, pulses, oilseeds, and several other crops. Thank you. This is a brief about the metroglyph analysis. Thank you. Please subscribe our channel. For different analysis models, please see our different videos like D-square statistics, like others. Thank you.